Okay, welcome back. So this is question 54 from biology section in IMAT 2012 past paper. So I will be answering this question. So it says, which of the following crosses is most likely to produce offspring of genotype G, small g, and capital N and small n. So basically the heterozygous for two genes. So basically, this question is about the crossbreed, specifically about dihybrid crossbreed. Okay, so it's a dihybrid crossbreed. So in this dihybrid crossbreed, so the thing is that it involves a two gene. So as we are shown in this example, so this is heterozygous of two genes. So we have one the heterozygous for the letter G and the heterozygous for letter N. So we have different alleles over here. One is being the dominant and one is being the recessive, and also same for this one. So it's a heterozygous. And that means we have one copy. We must have one copy of a dominant and one copy of recessive for both of the gene. So the simple definition about the dihybrid crossbreed is that there's involvement of two genes with each having two different alleles. So that's the most crucial thing about the crossbreeding between the two species. If you're focused about the genes, okay, so there are two genes involved, and two genes are being crossbred with two genes, so they produce certain offsprings also having the two genes. Okay, so now the most important assumptions that we must make about this question is that these are the key assumptions before we uh, go deep into this question, because they're very, very important. Without this, you might make a lot of mistake. Okay, so the main key assumption that we first must know is that the genes, the genes in this question, we assume ourselves in the most of this type of question is that the gene assort independently, independent. Okay, so the gene assort independent, and the most important thing that you must assume is that the genes are unlinked. Okay, unlinked gene, unlinked gene. So with what this means is that the genes are not close together, or not close together in same chromosome. Okay, so now to picture this clearly of what this means is like, for example, now this is the clear diagram I might have. So this is one chromosome I have, okay, and okay, so this is the one chromosome that I have over here, and then I have the another chromosome that is its homologous pair, and then I have the other chromosome which is a non-homologous. Okay, so it's non-homologous to that the chromosome above I'm showing you. Okay, so okay, so now this is the quick diagram about the this unlinked gene and what the definition about the unlinked gene really means. Okay, so now suppose this is the father's gamete. Let's say this is father himself, a father and individual himself. So this is a father's genotype. Okay, so I'm just showing you the picture about how the unlinked genes is supposed to mean. Like, So this is the father's chromosomes, okay, the one that's going to be crossbred with the mate to produce these organism. Okay, so now these two pairs are homologous chromosomes to each other. So we'll call this as a homolog. I will write a small short form. Okay, homologous pair one, and then the second one is a homologous pair two. Okay, so these two chromosomes are homologous to each other, and these two are homologous to each other. But these chromosomes are not homologous to each other. This one and this one. Okay. So yeah, so now we will list the genes of each and the alleles. Okay, so now let's say that this chromosome carries a gene for the G, okay, the letter G, and this chromosome over here carries, okay, like that. Wait, over here since this is a sister chromatid, so we can write over here like that. All right, so the other side, what we have over here is N. Okay, we have for the letter N, and then we have the dominant N, the recessive N, and recessive N over here. Okay, so these are the genes that are carried by each chromosome. So the most important thing we must also know that the two genes are carried on two different chromosomes. So there might be some confusion that most people might make is that they might think that like we have this small chromosome. Wait, okay. So now the most common mistake that some people might make is that, for example, I have this chromosome over here, and then I have the second one. Okay, so I just might cancel this out. Okay, so the common mistake that you might have the confusion in the exam is that some people think that there are two genes being carried by a single chromosome. So, like this, for example, I have dominant gene in this chromosome. I have the dominant gene in the other chromosome. Oh no, sorry, recessive gene. And yeah, the most important thing you must know is that the one chromosome cannot inherit two allele for the same gene. So every chromosome have different allele for the specific gene. Okay, so it cannot have two alleles for the same gene. So every chromosome has different genes. Okay, so different gene for a allele. Okay, so that's the main thing for specific allele. So and the other one is we can have this gene over here, and then we have N and N. So this is an example of linked gene. Okay, so these gene that you are seeing here are inherited together. So this is where the likelihood of crossing over occurs. But still, if the gap between them is very small, the most likelihood is they're being inherited together. Okay, but for this specific question, we just do not assume this at all. So this is the correct model. We must assume that the genes are unlinked. So this is the example of unlinked gene. So different chromosome carries a different gene. Okay, so each one have the different alleles also. Okay, so this is the example you must know. So this could be a father's genotype himself, or it could be a mother as an individual themselves. Okay, so this is the example you must know is that it's an unlinked gene. Okay, so the unlinked gene is the one that's going to be crossed over. Okay, so now the other one that we must know, the most easiest one is that each the dominant allele has over complete dominance. They're complete dominance, so meaning that the if they have a dominant allele inherited, so they will most certainly express, okay, over the recessive allele. Unless if you inherit two recessive allele, then you might express those recessive trait, but for dominant trait, at least one copy of the dominant allele will express the dominant trait. Okay, so now the other one is that gamete. as I've explained this also before, the gamete carry one allele for specific gene. Okay, so gametes must carry one allele for specific gene. Okay, so it's not like they can carry two alleles for same gene, so that's wrong. That's not also biologically correct. So every chromosome carries only one allele for a specific gene. Okay, so now this is the very quick example about the question about the thing you must assume, and also I'm going to quickly draw the diagram about the father's and mother's genotype. So this is a father and the mother's genotype. Okay, so mother's genotype, and then we have the first pair of the chromosome. Okay, the homologous chromosome. So have a genotype. It could be a gene G or the other one small g, and then the second pair we have here is N and then N, and then same for the mother herself. The first pair is this one. Second pair is this one. But you can change. Okay, so the it's not necessary the first pair must be the G or G genes. You can also write N gene in the first pair, but it doesn't matter. So I've set my parameters like this, but it's your wish. But the thing is, if you're setting parameters like that, then it must be the same for the entire process, or else it might go wrong. Okay, so now this is the thing we must assume is that gene assort independently and the genes are unlinked. Okay, so the most important thing is that there's unlinked gene, although the question does not specify whether the genes are linked or not, but in the most questions related about the gene and the crossbreed, unless they specify if there's a linked gene where the chromosome carries two genes, then you must always assume it's unlinked gene. Okay, so now moving on forwards. So now I'm going to draw a simple diagram about the gametes produced by each individual. So this is a father. This could be a father or a mother. Okay, mother themselves. So meaning this is the gametes of a father and mother that will produce in their body that would fertilize to produce the offspring. Okay, so now this could be either a sperm or it could be an egg. So and the so now let's move forward. So now first example, now these are the chromosomes I'm drawing here. The X, these are chromosomes. Okay, so now the first this pair, okay, G and then small g, as I've explained it before. Okay, and these are the other homologous pairs, N, N. Okay, this is the second pair and this is the first pair. Okay, so now this is a parent cell. Okay, so now the most important thing is that this is a first orientation. Okay, so what does this mean is that first orientation means that this is the first uh, possibility that it might produce the gametes. Okay, so I'm going to draw a diagram so that you can understand why I've written the orientation. Okay, so this cell undergoes meiosis 1. Okay, meiosis 1. Since it's a gamete production, so you must keep in mind it's a meiosis, not mitosis. Okay. So in the meiosis one, it produces two daughter cell, but they're not genetically identical because they do not have the chromosomes separated in a way done in the mitosis. So it is not genetically identical daughter cell, but we would say it's a genetically different daughter cell. Okay, so we have this chromosome, one chromosome. So these are not homologous pairs to each other. So this is a chromosome G that I've mentioned here, and this is a chromosome that's carrying the dominant allele L, N. Okay, so the other one is this chromosome. I have this chromosome and the other one, the small one, and they're non-homologous to each other. And then this chromosome is carrier. So this daughter cell carries a recessive alleles for the gene, and this one carries the dominant alleles for that gene. Okay, so the dominant G and dominant N, this one is carrying the entirely a recessive. Okay, so now these each daughter cell undergoes meiosis 2. Okay, meiosis 2. So when it undergoes meiosis 2, it will produce two gametes. So keep in mind that each of them produce two gametes. Okay, so that's fairly simple thing to know. So I'll have one chromosome like this. Okay, I'm going to draw more clear like that. Just a single unpaired chromosome, and it's also not unduplicated chromosome, just a single chromatid of each chromosome. So this chromosome is the one carrying the gene G, and the other one the gene N. So two of these gametes are produced. Now these are gametes, so this could be either a sperm or it could be an egg. Okay, in the female and the sperm in the male. Okay, so now the other one I have here is the other gametes that carries recessive alleles for the gene. So small g and small n. Okay, so now the genotypes of these gametes that we have here is the GN and GN. So this means that for this first orientation, their independent assortment, the most important one, is arranged in such a way that it produces these gametes with these genotypes. Okay, so their alignment is different. Okay, so now I'll explain about the second orientation. Now this will be a second orientation. Okay, so in this second orientation, I have the exact same chromosome. Okay, so the exact same chromosome carrying the exact same gene as this same gene and alleles as this the first orientation. So I have this GG and then small g, they're homologous to each other. And then the other one, I have this chromosome that carries gene for N. Okay, so this is the parent cell that will undergo the meiosis. Okay, so now it will go under meiosis 1. Okay, meiosis 1. So this will produce two daughter cells. Okay, so two daughter cell, and then the other one, two daughter cell. Okay, so likewise, as I've explained before, that each one has no homologous pairs in this, because in meiosis they undergo the homologous pairing where the crossing over take place and independent assortment. But in this question, we assume that there's no crossing over. But even if you set the parameters in such a way that crossing over occurs, it still won't make any difference because at the end you would get the exact same thing. Okay, so unless if you have a situation where the chromosome carries two genes, a single chromosome, then there might be a difference. But for this question, even if you assume there's a crossing over, it won't make any difference. You would get the exact same genotype. Okay, so now the arrangement for the second orientation, the arrangement might be different. Okay, so it would be uh, most likely it would be a heterozygous genotype. Okay, so why is that? Is because in previously we had daughter cells. Each daughter cell carries either completely dominant alleles and the other one completely carries recessive. But this time we have different. So this time we have one chromosome carrying for the dominant allele, the other one carrying for the recessive, and similarly, one is carrying for the recessive and one other one is carrying a dominant for the specific gene. Okay, so now this is the different combination that we're having over here. So now we will go forward. So to produce this will undergo meiosis too. So this will produce two gametes. Okay, so similarly as the first orientation, so we have this chromosomes like this, non-homologous to each other. So this one would be carrying the G and then small n, one dominant allele for specific gene and one recessive allele for specific gene. Okay, so now the other one I have here is recessive allele for G and the dominant allele for the N. Okay, so now these are the two gametes. So their genotype is going to be this and this. Okay, so now these are the two orientation that produces these overall gametes. So we know that in each orientation there are four gametes, but the two in each gamete are same. So meaning you will have two capital G and two capital N. Okay, because they're twice, because each daughter cell produces two gametes. Okay, so their genotype is going to be the same. So that's why there's two orientations to produce different gametes of different genotype. So now the overall genotype from gametes. So the overall genotype of from the gametes are four. Okay, are four of them. So those four of them are the these four gametes. Okay, so these are the four gametes that we have produced, and now these gametes are produced by both the mother and the father. Okay, so this mother and father produces this genotype because the question clearly mentions that there's a crossbreed between either it could be of two genes. Okay, so there's a crossbreed of two genes. So now we'll move on forward to draw a table. Okay, so the table where these are gametes of mother and father crossbreed to produce the offsprings of that certain genotypes, and they might express certain phenotype as well. Okay, so now this is the table, the father. Okay, so this is the father's gamete, or you can write a mother's gamete. I'm going to set like that. And then I have GN. The other one is small g and then GN. Okay, so similarly, and this is a mother. Okay, so this is mother's genotype and the father's genotype of their gametes. Okay, so now I'm going to crossbreed. So you simply just so GN, and then you multiply. You just crossbreed. You get this, and then you get that. Okay, so now these are the genotypes of the offspring produced by these parents with these genotypes. Okay, so now what we can do is that we can list out their certain phenotypes, their certain traits produced by these certain genotypes. Okay, so now we know that we have 16 offsprings produced. 16 offspring, and each has and overall there are nine different nine different genotype. Okay, so there's nine different genotypes. Um, if you count and search, and then you should ignore the one that are similar. Okay, so because like for example, this is the one that we have, the heterozygous of the two genes, and then I have the second one that is same as this. So that's why there are nine different genotypes from this 16 total offsprings, 16 possible offspring. Okay, so now I'm going to list all the phenotypes. So what does phenotype mean? It could be dominant or recessive traits. Okay, so the first one is going to be all dominants. Okay, all the dominant for of the two genes. Okay, so I have dominant so I have the dominant wait. Okay, so the dominant G like that. So then I have the N. Okay, so now if you count, there are over nine of these you might find. Okay, so this means that the D. Okay, so I'm going to draw here quick diagram. So if you see this symbol that I'm drawing here, this means this is a dominant trait. Okay, so it could be either a homozygous dominant or it could be heterozygous dominant. And similarly for N, it is same thing. Okay, like that. So this must be the, if you find such type of genotypes across this table, so it must be categorized in this symbol. Okay, and then for recessive, it's just going to be a small double G, and then it's just a recessive, and then same for N, it's just the recessive. Okay, so these are dominant, and these are the recessives. Okay, so you you will find nine of these genotypes, no, of these phenotype trait. If you look up for the certain genotype that has dominant G and the dominant N. Okay, so they're dominant for two genes. Okay, so the other one is just one dominant, and the other one is the recessive gene. So you will find three of these if you count. So let's count. I'll show the example. Okay, so if I count over here, so this is, yeah, one, and then the other one, if you find, then I have the second one here, and the third one. So these are the three, because there must be one dominant allele of dominant gene. Okay, and the other one must be the recessive gene. Okay, so it's a two gene crossbreed. Okay, so the other one we will have is the small one. Okay, so GG, and then the N will be dominant, and then we have three of these, and then the fourth one, the fourth phenotype is it's all recessive. Okay, so two recessive genes. Okay, so these you might only find one. So the only one that you will find is this right in the corner. That's all. Okay, so these are the phenotype traits produced by this genotypes of the parents. Okay, so now answering to the main question. So this entire working was actually just for the understanding. It's not actually for recommended to use this in IMAT. This is just introduction about this type of question for people who might not be aware about such type of question. Okay, so now the question says which of the following cross is most likely to produce offspring of genotype, this genotype. Okay, the one that has dominant, the two genes are dominant. Okay, so now what happens is that this example that I've used was the example for this option E. Okay, so the dominant, the heterozygotes dihybrid crossbreed. Okay, so now in that case, what we can do is that now to solve this question, what we'll do is just we'll focus on finding that particular genotype. Okay, so the probability of that genotype that they're asking, the heterozygote genotype, is this one. Okay, so the heterozygous gene for that one and the heterozygous for that gene. So now we will look for this particular genotype in this table. Okay, so now just going to erase this off. Okay, so now if I'm going to count, I might have four of them. So one, so this is the one genotype I found. Two, three, four. So there are only four of them. Okay, so four, and out of there are 16 offsprings. Okay, so this means 4 divided by 16. So this will give me 1 by 4. So this has very less chance. Okay, so there's only 1 by 4 chance of having the offspring with this genotype. So that's almost approximately 0.25. Okay, so but I'm not yet sure about the overall percentage of the other genotypes, so I'm going to solve that. But what I want to explain is that this entire working was just the explanation and intro for people who might not be aware. And this is the entire visualization about how you should visualize about the genes that must be unlinked. Okay, so I just have showed the example of how unlinked genes supposed to look like, and there's certain orientation that produces these gametes. Okay, so but for the IMAT, you must not do this working at all. Okay, so now this entire thing was just the brief explanation about how this question must be visualized, but now I'm going to show you the actual method that is recommended for IMAT. Okay, so now let's get back to question since I've explained much to understand about this question. So now getting back to question. Okay, now it says, okay, in the IMAT when you sit and you see such type of question. So first of all, they say the genotype of this heterozygote gene. Okay, the offspring that has this certain genotype. So they have the crossbreeds of these parents that crossbreed of their genotype. So now I'm going to go with option E. That seems the most basic. Okay, let's go with option A. Okay, so now the option A is for the option A. So this is the main method that I would highly recommend for the IMAT exam since it's very time crucial. Okay, so this is for option A, where there's this genotype crossbreed of the parent. Okay, so and okay, so like I've drew this table diagram. This is also very correct. You can easily find count the numbers, but it's a lot time consuming. So this is very time efficient. I'm going to explain. Okay, so now I'm going to separate the gene. Okay, since I know there's two gene crossbreed. So this is one, two, one, two. So I'm going to separate the first gene, which I'm going to consider as the gene for G. Okay, and the other one is going to be gene for N. Okay, so this is the first and the second. So now I'm going to focus on my first gene crossbreed. Okay, so that's going to be GG and GG. Okay, so this will produce four of heterozygote genes. Okay, so you can draw a Punnett square diagram. Okay, so like this, G and G. Okay, so this could be either father or mother. It's not specified, so you can set any ways you like. So this is how it is. Okay, so all of the offspring are heterozygous for this gene. Okay, so their genotype is heterozygous, so there are four of the heterozygous. Okay, so what this means is that since our question is focusing about the probability of producing this genotype, where I have heterozygote for both genes. So this means I'm focused about the GG. So since all of the offspring produces this heterozygote gene, this gene for G, okay, so this means 4 divided by 4. I have 4 offspring in this Punnett square diagram. So this will produce 1. So there's a very high likelihood, 100%, all of the offspring produces this genotype. So the probability of GG is 1. So this translates to being 100%. So this has a very high chance. And the other gene that I'm going to focus on is the the gene for N. Okay, N, and then the dominant, homozygous dominant crossbreed with the heterozygous dominant. Okay, so this produces the offspring with these certain genotypes. Okay, I'll have two heterozygous and two homozygous dominant. So this will give me, so since the question is about the heterozygous dominant, so this means there's a half chance. So why is that? Because I have two of the, I have two heterozygous only, so 2 divided by 4, this will simplify to half. Okay, so the probability of having this heterozygote uh, gene is half. So it's only 50% chance. So this translates to 50%. Okay, so only 50% chance. So this means if we combine the probability of having this heterozygous for two genes, so we'll multiply them. So 1 times half, this will give me probability as half. Okay, so I'm not yet sure, but I can make direct comparison that the option A has higher chance of producing that heterozygote of two genes compared to option E, where there's a crossbreed between G and N and then G and N. Okay, so this is very less chance of making it, but option A has higher chance than compared to option E. Okay, but I'm going to use this method and then show the which one is the greater. Okay, so the other part we have here. Okay, so now this will, as I've explained before, separate the two genes. Okay, and then crossbreed them individually. Okay, so this will produce two homozygous dominant and two heterozygous dominant. So the probability of having that heterozygous dominant for this gene is only 50% chance. And similarly for this NN, yeah, for this one it might be different. So it will produce one dominant homozygous and the two dominant heterozygous and just one recessive. So this will produce probability for heterozygous dominant is half. So the overall probability of heterozygous for two genes are, so this will be 1 by 4. So this is also very less chance compared to option A. So now the other one, we have this genotype crossbreed. Okay, so now then we'll multiply, we'll crossbreed this. So this will produce all the 4 heterozygous for this gene. So the probability of that will be 100%. And then the other one, this will produce 2. And then the probability will be of this heterozygous will be half. Okay, so the overall probability of having this or in her having the offspring of this genotype is going to be half. So it's still the half. Okay, so this is all. This might have higher chance, but this is similar to option A. So we can easily cancel these two out because we must find the one with the highest. And since you know these two have the exact same percentage, then in the exam, the quickest way is that you can cancel these out because there's only one option that you can choose, unless if the question says you can choose two options. Okay, but we'll get later to that. Okay, so now we have the other one. So this other one is quite interesting because it's the one crossbreed. There's a crossbreed where there's a two gene of homozygous dominant being crossbred with two gene of two recessive genes. Okay, recessive alleles. So now I'm going to separate, as I've mentioned. This will produce four of these. So the probability of this genotype is 1. So now similarly as for this one, so there's a 4. Oh, sorry. So there are 4 of these genotypes. So the probability of having this genotype is also 1. So this will give 100% for that. Also probability, so we just multiply 1 and 1, so this will give 1. So this is 100%. Now for option E that we've explained before, so this one is the most common heterozygous dihybrid crossbreed. Okay, so now we will, if we crossbreed each individual genes, so this will have 1 homozygous dominant, 2 heterozygous dominant, and just 1 recessive. So the probability of this one is just 50%. And same for this heterozygous dominant being crossbreeds. And then we have N, and we have 2 of these. And this will produce probability half. Half chance of having this certain genotype. So probability combined. So we just multiply together, we get 1/4. So this is very less chance as we've calculated before with different method. Okay, so now from all of these options, if we compare, the only one that has the highest chance of producing the offspring with this heterozygous gene for 2 genes is option D. This one is the highest. It has 100% chance of producing offspring with this genotype. Okay, so the only correct option is option D. Okay, so option D is correct. Okay, so just mark that out. Okay, so that's it for this question. So thank you guys for watching my video. I hope you find this video helpful. Please leave a like and subscribe to my channel. Thank you guys. Bye.